Today we will start with the DNA fingerprinting. DNA and fingerprinting. Why finger? Finger words comes fingerprinting. Before 1984, there was no science to detect whether the paternal identifications as a DNA level. But after the study of DNA fingerprinting, they called also typing of DNA or DNA typing. So DNA profiling is another word. Genetic fingerprinting, genotyping, identify testing in genetics, methods of isolation and identify variable elements within the base pair sequence of DNA. This is the definition. Simply you are telling that what mean by DNA fingerprinting. So what you will write? You will write, sir, it is a complete ATGC or nucleotide sequences of particular persons or particular genes or particular. But why this is happening? In many papers you will find DNA profiling. In many papers by genetic fingerprinting of so and so crops. So genotyping or identify testing as a genetic level that is including DNA fingerprinting. So differences in the base pair sequencing of DNA. This technique was developed by this Alex Zephyr, and they had noticed that certain sequences of highly variable DNA, that is known as microsatellites, and which do not contribute to the function of genes are repeatedly within the genes. Paul Hubbard coined the word in 2003 DNA fingerprinting form and alternately to conventional morphological based classification. It is now generally acknowledged approaches for determining genetic differences and relatedness. Whether they have taken a morphological data with their DNA data and they have conventionally classified according to that. But it is not a proven hypothesis. DNA fingerprinting is a technique that uses DNA markers to allocate breeding lines to heterocycle group to identify varieties. Molecular markers is a specific DNA sequence, or you can say variation in the DNA sequences is detected through that can use to identify the track genetic trait or a variation within the population or a variation within the individual organism also. These markers are often used in molecular biology, genetics, microbiology, plant and animal breedings also. So wherever you will find a genetics or isolation, that DNA markers are used, molecular markers are used, plant-based techniques. Breeders are also utilizing. Now, another terminology that is genetic polymorphism. Polymorphism means the same isolate or same DNA at same position or same location, whether it is present or absent. That I will show you in the gel also. So genetic polymorphism is classified or classically defined as simultaneously occurs of a traits in the same population of two or more discontinuous variants or genotypes. Although the DNA sequencing is straightforward approaches for identifying variation at locus, it is so expensive as well as laborious work also. DNA sequencing. So expensive that require a Illumina instrument more than 1 crores or 1.5 crores. Then solid sequencer. There are four generation sequencing, nanopore sequencing. So it is only, but in as I said in 1984, after 1985, this Jeffrey, Alex Jeffrey described the barcode like DNA fragment pattern that is generated by the multi-locus probes in the electrophoresis, separation of genomic DNA under gel. So recent terms, as you know, that DNA fingerprinting and DNA profiling that is being used, or combination of many versatile tools for various aspect of describe the various aspect of plant genomics and plant genome studies. So these include a characterization of the, as I said repeatedly three times, that genetic variability among the vegetable crops or among the flower crops, or a genome mapping can illustrated or taxonomically differences can be diagnosed through the DNA with fingerprinting. Now, properties of desirable for ideal DNA markers that can also understand for choosing which DNA marker. Can I use RABD? Can I use SSR? Can I use ISSR? Can I use RFLP? Can I use AFLP? These answer can be got from these. First, it must be highly polymorphic in nature. Whatever the markers you are utilizing, it must be highly polymorphic in nature. Second, now genetic terminology comes co-dominant inheritance. Once two terminology are there. One is homozygous, another is heterozygous. So heterozygous state of diploid organism or homozygous state of diploid organism. So whether it is governed by co-dominant inheritance or not, that you should understand, and then you apply whether these DNA markers can be used or not. And marker, before selecting marker, you have selected 10 decamer or 13mers. Whether this portion is present in that genome or not. So frequent occurrence of that genome can be possible, then you can choose that marker. Another, selective natural behavior, or you can say environmental condition or management practices of that particular desirable properties found in that or not. It must be easy or fast essay. It must be highly reproducibility of that markers. One time you do DNA fingerprinting in Lilium. I am doing in Lily. Another in also doing in the same species. But OPB markers you have used. Here is OPA marker here. I have also used OP series. And the fingerprinting is not reproducible. Not reproducible. You got a 8 bends in particular length. I got a 7 bends in particular length, and another got 6 bend in. How it is possible? So which marker you have selected, that is much more important. So easy exchange of data between the laboratories is also possible. Now types of molecular markers. There are two types of molecular markers utilized to evaluate DNA polymorphism. Generally classified as a hybrid based marker, or you can a polymer chain reactions based marker. Hybridization, one word. Hybridization based markers. And both the things are one, PCR is also used, another restriction enzyme is also used, and PCR based marker. Inform or DNA profile are visualized by hybridizing the restriction enzyme digested DNA to level probe, and which is DNA fragment to know the origin of the sequence. So nowadays we are classified into two types only, two types of marker, PCR based and non-PCR based. So those based on the PCR amplification of genomic DNA fragments, they are includes RAPD, AFLP, SCARS, STS, EST, single SNP, SSR, and caps. I will also ask the full form of that. The non-PCR based, that DNA hybridization between the RNA or DNA probe and total genomic DNA, generally there is restriction fragment length polymorphism, name itself indicate. VNTRS is also non-PCR based. I will explain in detail with the classification also. But you must know a differences between that and the full form of that. So first is RFLP. As I said that non-PCR based, restriction fragment length polymorphism. Then STS, sequence take sites. Then SSCP, single strand conformation polymorphism. Single strand conformation polymorphism. Then sequence take microsatellite site markers, STMS. Sequence take microsatellite site markers. Then inter simple sequence repeat markers. Inter simple sequence repeat markers, ISSR. SSR and ISSR generally used in detection or random primers. They are utilizing for DNA fingerprinting in many of the crop species. Okay. Then RAPD markers are also used for the DNA fingerprinting. Random amplified polymorphic DNA markers. Scar marker, sequence characterize amplified region. Remember, sequence characterize amplified region. Sequence characterize amplified region. 4. Now, up to that scar is completed. Sequence characterize amplified region. SCAR. For amplification of specific brand. For you have to write only these. Okay, scar. Cleaved amplified polymorphic sequence. Cleaved amplified polymorphic sequence, that is caps marker. AFLP, amplified fragment length polymorphism. So AFLP and RFLP, one is PCR based, this is non-PCR. Restriction endonuclease enzyme is used. So it do not require. Here AFLP, amplification you require. So PCR based. So many times they are giving the same differences also. We commonly use types of molecular marker. As I said, one by one, we will see next lecture what is the use of that and how it is used.